So good morning, everyone. Welcome to the first edition of Proteoform Thursdays. We're fortunate today to have Nick Young with us from Baylor College of Medicine. He's going to be speaking with us, but I just want to give a quick little overview. My name is Paul Danis. I'm with the Consortium for Top-Down Proteomics. And just give you a quick idea on what the consortium's about, and then we'll jump into Nick's presentation. So thank you for joining us. We have people from 20 plus countries. So good morning, good evening, good afternoon, wherever you are, and thank you for joining us. So the consortium has two main objectives. One is to help build the community. We do this through events such as this, workshops, education. We have a short course coming up at ASMS in 2021 this fall, a variety of other activities like that. The other is we try to guide the technology. We've developed some standards and methods. We have a variety of initiatives to try to develop good community awareness and assess the community and do things that can help raise the bar for the whole field. We have things such as initiatives in native top-down, monoclonal antibodies, protein separations, as well as protein standards. So we're trying to do a bunch of things on that side as well. And if you have ideas for other initiatives we should undertake, please feel free to ping us or contact us. There's a variety of resources for folks who are trying to get into the field or folks that are just looking for more information. At our website, if you don't want to type top-down proteomics, you can just type proteoforms.org. That'll get you there as well. We have a variety of methods, standard methods to start you out. We have a bibliography there. We have some publicly available software tools that we list there to help folks figure that out. And we have some other guides on notations as well as the Proteoform database. Also next, just a sneak peek, our next event will be May 20th. We're fortunate that Professor Si Wu from Oklahoma will be here to speak about our high throughput quantitative top-down proteomics. So we'll have more information on that going out next week as far as registration and details. So stay tuned for that. So if you haven't become a member yet, now it's currently free to join the consortium. If you go to the website, it'll take you about 20 seconds to sign up. And you'll be the first to hear about these kinds of events, as well as we'd love to hear from you as well about the kind of things you want to see us be involved with and engage with. So these wouldn't happen without the support from our fantastic sponsors. We want to thank Thermo Fisher, Bruker, SciEx, Agilent, Waters, as well as Pfizer and New Objective for providing the support to enable us to do the variety of initiatives as well as events such as this. So that said, let me hand it over to Nick. Nick is doing fantastic work around proteome-specific biology. He's going to tell us some of the latest findings from his group. Nick is the assistant professor at the Werner and Mars McLean Department of Biochemistry and Molecular Biology, as well as the assistant professor in the Department of Molecular and Cellular Biology. There's more details on his bio that was sent out when you registered as well, so please take a look at that. So Nick, I'll hand it over to you and go ahead. Just one other note is please jump in questions in Q&A, throw them in there, and the audience can, if you want to comment on other people's questions or you want to upvote questions, that's fine as well. So go ahead, Nick, it's all yours. Thank you.
Thank you, Paul, for the introduction. Let me get my screen set up and hopefully that will, yes, there we go. So that should be the full picture for you. So thank you for the introduction, Paul. I'm honored to give the first lecture in this new seminar series. I think Proteoform Thursdays will be a great platform for us to communicate as a community. And in that regard, I am going to be sort of focused more on some of the central questions and some of the methods that we all face before going into sort of how we can use the technology we've developed to really reveal mechanisms around transcription regulation through sort of this lens of quantitative proteoform biology. And please do feel free to interrupt, raise your hand, say that you'd like to talk. I believe Paul will be monitoring that, and you may be able to be unmuted and we can have a discussion in the middle. But towards the end, some nice robust discussion as well. So on the most sort of philosophical level, the question that we're asking is where does the complexity of life come from? And it's not through the size of the genome. Humans are actually sort of a very moderate sized genome, and onions have a larger genome than us. Rice has a smaller genome than us. But we actually have fewer protein coding genes than rice. And so this is one of the surprising results that like, well, how do we have more complexity if we have fewer genes? But we have actually a relatively large genome relative to the number of protein coding genes that we have. And so it is this sort of extra genomic material that is essential. And that's because this is playing a key role in regulating the genes that we do have. So it's not just the genes, the number of genes, but how you're using them in a very nuanced and very regulated way. And so it is this genome regulation that accounts for the complexity of life, allows for multicellular life through differentiation into different cell lineages, tissues, organs, and even on the whole organism level, the genome of each individual cell in an organism is finely tuned and controlled in a very specific manner. And so this extra genomic material is regulatory elements, promoters, enhancers, silencers, insulators. These are all key to having a sophisticated system of use of these genes. But that's just the cis-acting description of this, because there are significant trans components to this. And in fact, the genome in its physiological state exists as a chromosome, which is about 50% protein. And these proteins play a key role in this regulatory process. In particular, we focus on nucleosomes and the histones within them, and the post-translational modifications that serve as signaling platforms to regulate the genome. So the nucleosome consists of double-stranded DNA wrapped around an octameric core protein complex of histones that license and regulate access to this underlying DNA sequence through post-translational modifications on their N-termini. And there's a complex system of writers that write these PTMs, erasers that remove them, but also readers that retrieve this information from this genomically indexed information database, essentially. And so this system has actually been studied quite well for about 20, 25 years or so, but primarily using technologies that don't operate on the proteoform level. They operate on the individual PTM level, but nonetheless, this has actually shown us where these regulatory elements are, because they're not at all obvious or even predictable based on the DNA sequencing. And so the genomic localization of these PTMs tells us a lot about the genome, but not all PTMs make sense when you look at them in this way. And that's because this assumes that there's one PTM, one function, rather than one proteoform, one function. And so I argue strongly against this concept of that each PTM has this injective relationship with function. And in part, this is due to the fact that the readers that retrieve this information, they have these evolved domains that site-specifically recognize PTMs, but they're usually two or more of these on each of these reader proteins. And so they're reading combinations, they're reading proteoforms. Furthermore, histones typically don't exist with one PTM only. Maybe 12% have just one PTM. Most of the time, they're in a hyper-modified state that is best described and studied from a proteoform perspective. And so we really see the proteoform as a necessary expansion of the central dogma, that beyond protein, yes, you can regulate proteins with a PTM, turn them on, turn them off, maybe even give them different function. But there's combinations of post-translational modifications giving rise to many different proteoforms, each of which may have distinct function. And there are additional sources of variation, notably in histone, the presence of genomically encoded variants, because there are many different variants that do indeed have different function. So to study this system from this perspective, we of course need to use top-down proteomics. However, I argue strongly that it really must be quantitative. We must quantitate proteoform in order to study proteoform biology. It requires observation of change. You make a perturbation, you see a proteoform change. An enzyme comes in and writes more information. You need to observe change in the abundance of the proteoforms. And you need to do this for many hundreds of different proteoforms in an unbiased manner. But this is challenging. These are mostly structural isomers. That means that they have very similar physical characteristics under many types of chromatography. They co-elute. They have the same elemental composition. They're unresolvable in MS1 in the mass spectrometer. And so the solution to this problem, for us at least, is to achieve better chromatography, proteoform selective chromatography. So not necessarily the sharpest peaks, but the greatest separation of these structural isomers from one another so that we have minimized the amount of mixed spectra that we have. Nonetheless, we do still have mixed spectra, but the spectra are less mixed, and they're to the point where we can back calculate the contribution of like a binary or maybe ternary combination using mixed integer linear optimization mixture model. And we can do this quantitatively so that we can then pull these apart, put them in different bins, and study the proteoforms from this quantitative perspective. Of course, we always work in biological triplicates or more, be these cells or tissues, and we're usually taking multiple measurements over time or in response to different doses. So we're typically not doing a binary comparison between an on state and an off state or something like that. We're usually looking at different doses or progression of time. We're doing very narrow isolations to reduce the complexity of the spectra as well. And for histones, we're doing pretty much exclusively electron transfer dissociation. And then we interleave our technical triplicates. So we do these long runs where we are trying to separate those samples that are related to each other as distantly as possible in time so that we don't have batch or order effects. And so with this, we can get quantitation down to about 10 parts per million in abundance. This is highly reproducible. On biological replicates, weeks apart, we get Pearson correlations of 0.9. We've worked very hard on the getting from cells. And so our metrics of sensitivity is number of cells to data. And so we've gotten this down to about 2 to 3 million cells. It's a fairly high throughput and robust method that works every time, 24 hours a day, 7 days a week. And due in part to this high throughput, it is cost effective. And I would argue that it is more cost effective than doing a Western blot. It does require that you be unbiased and that you look at all PTMs, but you gain this additional information of extreme value, which is the binary combinations and the proteoforms. Now, I just want to take a second to back up and say, okay, well, this is linear and all of that, but there are still some questions. And I don't think it's obvious that we can do a good job quantitating proteoforms, even as a community. In part because, just identifying proteoforms is fundamentally challenging. There are not really great metrics for how good of a job we have done identifying a proteoform. You can reverse the sequence and that really doesn't tell you anything. We're actually quite confident in our protein identification to, I would say, a ridiculous degree that we're confident in our protein ID. It's simply the subtle localization of the PTMs. And so we're oftentimes off by just a little bit. And so to test this and to understand the power of the quantitation here, we need to do some null hypothesis testing. That is to say, what is our false discovery rate? Is it consistent with p-value based statistics? And so for this, we've done a couple of different things. One is inject the same sample 18 times in a row under the null hypothesis. So that's the technical null hypothesis. Or do 6 tissue culture dishes that have been treated the exact same way and do 3 technical replicates of each. And each of these create 18 samples, data sets that should have the same conclusion. And then we can group them and do t-tests and get p-values. But since these are identical, we can actually reorder these 1,000 different times and bootstrap this up to 169,000 p-values. And what this tells us is that when you bin these, you get an approximately flat distribution. And so this is actually, so there are a couple of different false discovery rates, not just in the bottom-up proteomics world, but there's more in statistics, there's a false discovery rate. And so this height here is really your false discovery rate. And when you have real bona fide non-random changes, you will have a peak down here because it will no longer be the null hypothesis. But under this null hypothesis, we get about 4.5% positives at less than p equals to 0.05. So this is approximately exactly what you would expect. Now we can take this a little bit further. It's actually a little biased towards fewer false discoveries than expected. But we can use these p-values even further. And one thing the first consortium project taught us is that near misses are quite common. So we're oftentimes almost right about our proteoforms. And so you will mix up related proteoforms more easily than distantly related proteoforms. And so we can then do correlations between these p-values. And so as you go through those different permutations, different bootstraps, you can see how the proteoforms correlate to one another. And if they are correlated, that tells you that you are making systematic errors here. And so then we can go in and identify what are the errors that we're making and correct them and remove those correlations. Another thing that this tells us is that the false positives are evenly distributed across abundance and are uncorrelated. So that is that a highly abundant proteoform, although we have a stronger sense of the identification, is just as prone to a false discovery on a quantitative level as a low abundance proteoform. It may have a smaller coefficient of variation, but you're not going to discover that it has changed any more frequently than the lower abundance proteoforms. Now, the lower abundance proteoforms are more common. So another question here that we need to answer is this accurate? Can we validate the ground's truth of such approaches? And so what we've done here to try to get at this is to take the same sample and simultaneously do top-down proteomics and middle-down proteomics on the same sample in the same LCMS run. So that is, we've partially digested the protein, and we have both the intact and the digested product, which contains actually all of the PTMs that we're looking at, and we can get complementary data. And for the most part, this agrees above 99%. And what both of these tell us actually is that not all proteoforms exist, and they're of very unequal abundance. So where just the combinatorics tells you that there might be trillions of different possibilities, the empirical data tells you there's hundreds, maybe 1,000. Okay, and actually this is important from an information theory perspective of that if these were all uniformly distributed, that says there's actually no information content here. And so because this is a system that is actually rich in information, there is this strong bias. There are hierarchies, there are dependencies. So what can we use this for? So one of the areas that we've been very interested in is therapeutics. Can we sort of learn how to treat disease more effectively? And so in the area of chromatin and epigenetics and gene regulation, we can, all of these readers and writers, and even the erasers are eminently druggable targets. And there are in fact a lot of drugs that are FDA approved or in clinical trials. And they're very effective against certain indications, but not in an obvious way. That is, this is a proteoform-based system. You're inhibiting an enzyme that you don't really understand what it does. You understand that it writes this PTM, but not really the context in terms of proteoforms. And so you can't connect the drug or really know how to use the drug if you don't understand the fundamental biochemistry of the system. And so one thing we can do here is look at the very short-term dynamics very early on, as soon as you give the drug. And this has not really been looked at that deeply before. And so in this case, we're just taking cells, we're dosing them, we're waiting just a few minutes rather than a day or 2 or 3 days. And the first thing we see here is that, as expected, we're inhibiting a deacetylase. We have an increase in acetylation. However, that acetylation is actually specific to the methylation state at another site, just a few amino acids away. And this is actually true for all of these different acetylations. And at the proteoform level, there's only these 3 proteoforms that are being increased. And so it is not that all acetylation is being increased. All acetylation is being increased, but not all proteoforms are increasing. And so there's a very specific hierarchy. You're starting with K20 dimethyl, you add K16 acetyl, K12 acetyl, K8 acetyl, K5 acetyl. And the other thing about these sort of early dynamics in sort of therapeutic applications is that as soon as you remove the drug, the cells recover to their exact epigenetic state that they started at within 30 minutes. And so this should not be surprising because it is epigenetics. That is, it's a persistent state of transcription of the cell. And so this says that, it's not through sort of like chemically slow changing post-translational modifications that epigenetic states are maintained. It's through active biochemical proteoform level mechanisms. And so these effects are transient. Another thing we can do with this technology is ask the questions in vitro. We saw these specificities in vivo, but do they recapitulate in a test tube? Usually when people are trying to understand what these enzymes are doing, they take recombinant substrate, they add this enzyme, cofactors, SAM, cell-CoA, etc., and look at what new products are created. What we do is we purify the physiological substrate as it exists in the cells, that is proteoforms. And we give a complex mixture of proteoforms that we've measured quantitatively to the enzyme, and we get new proteoforms. And so what this tells us is that indeed, even in vitro, these enzymes, through their active site alone, are not, they're generally described as being promiscuous because they write at so many different locations, but they only do it in a very specific order. That is, the proteoform substrate specificity is actually exceptionally high. This can also reveal drug resistance mechanisms. In this case, we're inhibiting a histone methyltransferase, sub-420, which actually writes this post-translational modification, histone H4 K20 dimethyl. And we do this in a couple of different cell lines, one of which is resistant to the effects of the drug at the PTM level, while the other one is prone. So you can see fairly quickly, within hours, you have a substantial decrease in this post-translational modification. However, taking a proteoform biology, quantitative top-down proteomics approach here, you can see that it's actually the resistant cells within 15 minutes of application of the drug are having a dramatic, broad, and early response, whereas the prone cells have a much slower response on the proteoform level. So if we drill down into this 15-minute time point, we can see that, and I'll remind you, K20-dimethyl is the PTM that this enzyme writes. And indeed, you see this dramatic decrease in this very specific proteoform that only contains K20-dimethyl in the constitutive N-terminal acetylation. But it's not because the methylation is going away. It's because this substrate is being acetylated, and you're having this shift from a hypoacetylated version to a hyperacetylated version. And so these are actually many of the same proteoforms that were being created when we added our histone deacetylase. And that is the histone acetyltransferases came in and wrote these. And so this, just looking at this system from a proteoform perspective, tells us when you inhibit this methyltransferase, yes, in many cases, the competition between the methyltransferase and the demethylase is shifted, and the demethylation occurs. But in resistant cells, and I'll remind you that cells have evolved mechanisms to resist these changes. They've seen these sorts of molecules before. And so they activate their histone acetyltransferases, and they acetylate specifically the proteoforms that contain K20-dimethyl. And now, this is no longer the substrate for the demethylase. Those flanking acetylations block that activity. And so now we know this evolved mechanism. And in order to affect the change that we're looking for, all we have to do is shut this down. And so we add a histone acetyltransferase inhibitor, and indeed, we're able to overcome the resistance. Now, I'm not going to show you all of the data, but we've actually worked out the system quite a bit further, that from monomethyl to dimethyl, you need to be in a hypoacetylated state. And from dimethyl, this is the preferred substrate of the histone acetyltransferases that takes us to this dimethyl hyperacetylated. And this is the preferred substrate of SMID3, which writes K20 trimethylation. So this will not write from dimethyl to trimethyl. It'll write from hyperacetylated di to hyperacetylated tri. And then this is readily deacetylated down to trimethyl. And so it's a long path to get from dimethyl to trimethyl. And the reason why is that this trimethyl is actually sort of this deep heterochromatin, and the dimethyl is much more active, or at least prone to activity, not sufficient. But this sort of shuts down transcription. And so if you want to have certain genes, like developmental genes, permanently shut down, you need to have this sort of tortuous path to turn them back on. And so we can use this actually to this sort of technology to inform effective therapies in combination with each other. And this is actually focused on the readers now. And so this is a mouse model, a PDX model of acute myeloid leukemia, looking at the bone marrow of wild type versus AML mice. And you can see that, again, you have these sort of hyperacetylated proteoforms. But when you look at them, you know, the acetylations are actually quite similar in abundance. There's nothing really critical to learn there by the sort of the level of the individual PTMs. But when you look at the proteoforms, these are not the same proteoforms as with the histone deacetylases. These are proteoforms that have these sorts of out of order, missing acetylations. And these sort of combinations like 5 and 12, 8, 16, these are good combinations that are known to be read by proteins such as BRD4. And so we have compounds to both sort of shut down overall acetylation through metabolic pathways above this, as well as the ability to shut down the reading of these sorts of proteoforms by BRD4. However, shutting down AMPK alone does not cause increased mean-free survival, and nor does shutting down the BRD4 recognition of these proteoforms. However, when we use these in combination with each other, we get an essentially twofold increase in mean-free survival, or 1.5, I would say. Recently, we've been going quite a bit into proteoform biology and sort of neuroproteoform biology. And we've actually mapped out the proteoforms of 11 different regions of the brain. We can sort of drill this down to individual PTMs and how they are distributed in different portions of the brain, inferring sort of different levels of transcriptional activity. And these sorts of silencing marks oftentimes are well correlated with each other spatially. But we're focused primarily on aging and diseases of aging. And so this is just our preliminary study on mouse brain aging. And when you look at the PTM level, you see relatively few changes that are both statistically significant and more than twofold. And one thing I should point out is that our error bars, that is our precision on these measurements are quite high, because we are taking so many spectra, both multiple replicates, but also these are sort of marginalized out of proteoforms. And so we have actually quite good precision and statistics, but many of these are not changing twofold, but they're still meaningful. But when you look at binary combinations, you can see that there's far more specificity to these, and there's far more to learn from them. And in fact, some of these binary combinations have been previously described as being age associated. And so this one is highly statistically significant. We can measure its distribution within the brain as well. This binary combination, which is both the most statistically significant and the largest fold increase, p less than 0.001, but the PTMs themselves are either changing with far less statistical significance or not changing at all. And again, we can localize this within the brain. And both of these aging-related binary combinations have similar distributions, notably high in the cerebellum, where a lot of age-related neurological diseases initiate. Now, backing all the way out to the full proteoform level, and I would note that we're mapping the absolute change in proteoforms by the size of the circles, in addition to having the fold change on the x-axis and the p-value on the y-axis. And this is important because, as we saw earlier, sometimes change is not twofold, but it might change, 40% of the genome. If you're changing 40% of the genome, but it's not a twofold change, you're going from like 40% to 80% or 79%, you're not hitting that twofold mark, but you changed almost half of the genome. So it matters a lot. And so some of these proteoforms are changing, 10%. That's huge. And so, but mostly these are the common proteoforms. Those in the top 10, the top 50, these are the ones that are decreasing with age. And you have these many small increases in these odd proteoforms that are really non-existent in younger mice. And so this is sort of indicative of a general dysregulation of this proteoform biology. And so currently we're moving into Alzheimer's models of mice with brain region and cell type specificity. And we're combining this quantitative proteoform biology with genetic manipulation of mice. That is, Kree-Lox mice turning on and off genes, AAV expression into specific cell types within the brain that actually have phenotype that we can observe, longer life, for example. We're also looking at the transcription factors and their proteoforms upstream of some of the processes that we're interested in, primarily around lysosomal biogenesis. And then measuring the downstream effects by doing lysosomal proteomics, so we can pull out intact lysosomes out of living mice that have been genetically manipulated. And we have looked at sort of the quantitative proteoform biology of their epigenetics. And with that, thank you for your attention and for all of the people that did the work: Bethany Taylor, Faith Joseph, Carl Pancha, previous graduate student Matt Holt, and previous postdoc Tao Wong, and funding agencies, National Institute of General Medical Science for my primary R01, and also support from the National Cancer Institute. And I would love to have an extensive discussion and answer all questions now.
Great, thank you, Nick. Fantastic stuff. We have a couple of questions to get us started, and if other folks have comments or questions, you can either raise your hand or throw something in the Q&A here, or comment on someone else's question if you like. So, Nick, you want to just go through and read out the first question and go ahead from there?
Sure, yeah, I'd like to encourage people to be able to speak as well, if you can unmute them so they can have the conversation. But so how do enzymatic versus non-enzymatic PTMs fit into the proteoform model? I guess the later could be equated to denote mutations of proteins. We do actually have oxidation, for example, is actually quite common in some of these proteoforms. And some of that is actually maybe meaningful in the biology, but a lot of it is artifactual. I think you're referring to the fact that if you simply put, acetyl-CoA in high concentrations with a protein, it's gonna acetylate its lysines even without an enzyme. And so I guess I would see that as being part of the system. That is, that you will have these non-enzymatic kind of spurious PTMs that aren't enzymatically driven, but are metabolically driven. That is, if you have an abundance of certain metabolites that are necessary for these chemical reactions to occur, they'll occur, and they're actually signaling in information about the metabolic state and sort of the environment of the cell. So it's not a mistake that acetyl-CoA is part of the TCA cycle, part of central metabolism, and it also is a signal to say turn genes on, because I have lots of energy to produce proteins. Hopefully that answered that. Perhaps we could unmute everybody so that they can talk. All right, and let's see. All right, thank you for the very interesting talk. Could you again elaborate on how different proteoforms can be distinguished by mixed integer linear programming? So this goes to the mixture. So that is the, frequently in proteomics, the question is framed as being, I have a spectrum, what is the identity? And so I think that is a mistake, because there is inevitably more than one thing there. Most of the time, the other things that are there are not interesting, unrelated, maybe not producing significant fragments. But in the case of proteoform analysis, it's frequent that what's there is an isomer of the proteoform, of the primary proteoform. And so you have a proteoform that is dominating the spectrum, or maybe sharing the spectrum, but there's another proteoform there. And so if you look carefully at your fragment ions, you can see that there's sort of a divergence. You might have fragments, and then they'll diverge into different modified states of your C6, C7, C8 ion, for example, and then they'll come back together. And that says that there is more than one proteoform there. And so you can back calculate that. And you can do that with linear programming. You can do it very fast, and you can reach optimality for whatever objective function you give it. So whatever you tell it is sort of the best fit, it will achieve that very, very quickly. And so you can punish the use of excessive numbers of proteoforms. It doesn't tell you the absolute truth because it's dependent upon the objective function that you set up, but it does do that quite effectively.
Nick, maybe if we go to on the Q&A, there's one that seems to have some interest here. Is how do you see the utility of ion mobility for proteoform separations prior to MS?
Okay, I was just looking in the chat. All right, so Lloyd, absolutely. Now there's some challenges there. Ion mobility can help solve exactly what we were talking about, of that if you can further pull apart those isomeric proteoforms from one another, just like we're doing in our proteoform selective chromatography, you have fewer of these problems of mixed spectra. Now that the challenge there that I see, however, is maintaining sensitivity. And so we do need to maintain, and so there are ion mobility approaches that are concentrating. And so you can, rather than dispersive. And so you need to maintain sensitivity while achieving this higher selectivity. But yeah, I think there absolutely is a future there. I'm not sure that there is an absolute solution that is optimized for this problem at the moment. So what does it mean by quantitation, quantitative in this talk or study? And why is it important for proteoform studies? Okay, so by quantitation and quantitative, I specifically mean not quantify or quantification. That is, I'm defining this as knowing both the amount and the variance in the amount. So that is, we have a metric of precision, and that is essential to being able to determine if our hypothesis is likely to be correct. So I'm not quite sure. So it is important because, as I stated, we need to observe change. And so all of biology is based upon not identifying, but seeing differences. And so if we can't see differences between states, we can't determine function, and we can't reveal mechanism. All right, Jeff, great talk, Nick. I really enjoyed the proteoform phenotype correlation you were making. Okay, thank you. Abhishek, thanks for the talk, Nick. In the quantitation, do you see the potential for electrophoresis separation for proteoform separations? Yeah, no, I think absolutely. For like glycosylation, I think that works exceptionally well. Some of the methylation and acetylation, it's a little bit harder to achieve that with electrophoresis, because it's not necessarily having that large of a change and so there has been some work in the past where, with the right coatings and additives, you can get good separation of proteoforms. Unfortunately, they're generally not mass spec compatible, or they haven't been developed to that level yet. All right. All right, Maowei, great talk. Sorry, I did not fully catch the beginning of your talk. What are your general recommendations for the minimum number of technical replicates, biological replicates, for getting good statistics? Okay, so you absolutely positively need 3 biological replicates. There's no doubt about that. I generally go for 4, in part because if you end up with 3, you're okay. I think more is definitely better. 4, 5 is good, but yeah, minimum 3 technical replicates we do a minimum of 2 technical replicates, because that if they disagree with each other, you can't do statistics on that, but we're not interested in doing statistics. But if they disagree with each other, then you have evidence that there's something wrong, and you have reason to investigate. So, Susmita, how ETD is helpful over HCD and proteoform quantitation in bottom-up proteomics-based PTMI. We found HCD has given more coverage. So this is highly dependent upon the protein, and ETD is not at all the most common or the easiest fragmentation method. It's highly, it kind of needs to be optimized for each protein or each peptide. There are ways to sort of estimate what the reaction time should be. They're not extremely good. Histones are the most basic proteins that there are in the human proteome. And ETD is very good at more highly charged species. It is useful in top-down proteomics in general, but for this specific application, it is outstanding. All right, Julian, a disadvantage of MS is that it relies upon a population, say 1 million molecules. Do the statistical methods you use address this issue? So I think, Julian, this goes to a couple of things. One of which is that when we are identifying a single molecule, a proteoform, we do have a population of proteoforms that should be identical. There are, of course, limits in sensitivity, and as we go lower in sensitivity, we have lower in abundance. We have issues with we make more mistakes in identification. And so it's a limit. We could definitely use more sensitivity. I don't think it really affects the statistical methods that we're using. I think that we're definitely, however, losing information at those lower levels. And it is a motivation to have the better proteoform selective chromatography as well, so that we don't suffer ionization suppression and we're able to properly sample. The more separation that we have, I think the more accurate our quantitation is. But it is reproducible, though. All right, Joe Liu, are you pretty much set on your TDP technology platform? Are you considering other methods? There are methods that we do explore. Electrophoresis. We've done a little bit of work trying to see if that can work well. I think the ion mobility component, I think, could really be a big advantage if we had something like a TIMS device in front of what we're doing, in between our chromatography. It's not really super compatible with an Orbitrap, but we would need electron transfer dissociation in an ion with ion mobility and high resolution at the end, and that's not quite here, but I think it's pretty close. So yeah, definitely considering that. All right, James Fulcher, great talk. A couple questions. How are you dealing with missing values in the quantification? Also in your volcano plots, why are there different proteoforms aligning at the same log 2-fold change, but with different p-values? All right, so we are imputing. That is that, and we are also removing those proteoforms that are not reproducibly observed across biological replicates. And so we require that the proteoform be observed in at least 50% of the biological replicates. And then for 0 values, that is when there are no, when it is not observed in one condition and it is observed in another condition, we can't divide by 0, obviously, right? And so there, that becomes infinite increase, and we set that as a fixed value in our volcano plots to indicate an infinite increase or decrease. So, Jerome Gint, are there specific parameters you use to validate your proteoforms, or do you check each one manually? Yeah, so we do a lot of manual checking. We use some of the p-value correlations to try to identify the proteoforms that we're consistently making mistakes on. I would say that it's important to understand that, it's easy to validate the high abundant proteoforms. Many of the low abundant proteoforms will validate as well, and we do that manually. We know that there are mistakes, especially in those lower abundance proteoforms, but we're assigning them very, very low abundance values. And then we're looking for statistically significant increases. And so there's a difference between validating identity and validating quantitative changes. And so there's different approaches here. You can validate the identity before you do the quantitation, or you can look for the statistically significant change and then validate the identity. And so it's not infrequent that we will have a very low abundance in one state for a particular proteoform and then a very high abundance in another state. And we will be able to validate the high abundance, but the data on the low abundance is much more murky. So, all right, Liliana, awesome talk. Wondering how would you go about linking specific proteoform to specific DNA region? Yeah, so this is a pipe dream of mine that I've thought about for a decade or so. And we have done a little bit of work here. And I would say in some ways we're not achieving what we would like to achieve, but we are doing just as good or better than what everybody else is doing. That is, so there is ChIP-seq technology. You use an antibody to pull down a portion of the genome that contains your post-translational modification. Okay. And that's like on base pair resolution and all of that. What they don't have is proteoforms, but they know exactly where those PTMs are. Okay. What we would really like to achieve is base pair by base pair, knowing where every proteoform is. But what we can achieve is that we can do a ChIP-seq and ChIP mass spec experiment at the same time. So we can pull down against a particular PTM, we can do the sequencing, and we can pull down against that same PTM and do quantitative top-down proteomics. And then we know all of the proteoforms that are at the locations where that are mapped by the ChIP-seq. So it doesn't tell you where each proteoform is, but it tells you which proteoforms are there. And when you do that sort of experiment, you actually get really surprising results. So like if you pull down against H3K4 trimethylation, you get a bunch of proteoforms that have all sorts of other PTMs there that are highly specific. And it's a very narrow band of proteoforms that you're looking at. But yeah, that's the best we have achieved so far. You could do sequential. There's other ones where we are actually going to tag proteoforms that are that have been proteolyzed. So we have truncated versions of these proteins that exist in vivo, and then we just express them, and then we can tag them and pull out where are they in the genome. So not perfect, but yeah. So what's the maximum distance, sequence, or spatial of dependent PTMs you have seen? Maximum distance. All right. So I assume you mean in the sequence. There's a high level of dependency between post-translational modifications that are pretty far apart from each other. We don't see a whole lot of correlation there. I think that the correlations that are there, like nearer proteoforms have more dependency on each other. There's definitely a little bit of truth there, but that's somewhat of an artifact of what people have been capable of observing. So we see, like, for example, a high dependency on K4 trimethylation and K27 acetylation. Those are pretty distant from each other. And then I guess spatially, I mean, the ChIP-seq data says there's lots of sort of spatial relationships there, but on the nucleosome level, we see a lot of correlations in the composition of the nucleosome. And so that is, we're able to pull out individual nucleosomes with all of the histones that are there, and look at what sort of co-purifies. And the composition of nucleosomes are really well correlated. So across, the variant that is present, for one of the histones versus the post-translational modification state of another histone in the same nucleosome, those are highly correlated as well. So in general, I would say correlation is more the rule than they're not being related. What drives all of that is another question. Part of it is the proteoform specificity of the enzyme, but a lot of it is the three-dimensional organization of the genome in the cell, and what enzymes are available, and what histone variants are there, and what histone chaperones are there, et cetera. So, yeah.
That's great. That's fantastic, Nick. I think we'll wrap up here. Nick, I want to thank you very much for this totally fascinating seminar. This proteome specificity seems like an emerging area, and I kind of wonder if all these problems that have been in drug development are because they don't have this. So maybe someday they'll get around this issue.
I think so, and I think in general, our field has a huge amount to contribute here. Not just in the area of biology that I'm focused on, but like the enzymes, they are working on proteoforms. Proteoforms are what are present in cells. That's what's there. And if there's a PTM there, we need to be studying it separately. And so, there's a fundamental aspect to drug development that's really, really being missed here that I think we can contribute to as a community. So
yeah, it's fascinating. It's gonna be great to see how this evolves in the next few years. So thanks again, Nick. It's a tremendous way to kick us off here. And I want to thank everyone for attending. And don't forget, next seminar, May 20th, we have Si Wu will be speaking with us, and we look forward to that as well. So thanks again, Nick, and I wish everyone a happy Earth Day, and we'll see you next month. Bye-bye.
All right. See you next month. Look forward to the talk.
Bye-bye, everyone.